Thank you, Carol. That's really inspiring and touching, and thank you for your support for all the legacy of excellent research education here. So now I'd like to announce the next, our speaker, Archimedes Lecturer, Alan Kaganov, Professor of Biomedical Engineering. I think after his lecture, Ke Cheng will answer a question. But first, let me introduce Professor Ke Cheng. And Professor Cheng joined our faculty this fall, and after he joined, he was already brought his student from other location, other university, bring a wonderful and vibrant group here. Professor Chen's research expertise is in pulmonary bioengineering, as Carol mentioned, which is of really increasing importance and relevance to the society since the COVID pandemic. His research is critical to global health needs and capture really our ethos and the spirits of engineering for healthy humanity. Ke Cheng earned his Bachelor of Science degree in pharmaceutical engineering at Zhejiang University in China, and earned his PhD from biological engineering from University of Georgia. He also received the prestigious, recently, Walter Coulter Award for Healthcare Innovation. So I'd like to invite Ke Cheng to the stage, and we can celebrate his professorship, and after that, he will give our lecture later. Ke Cheng, please join us.
Thank you. Good evening, everybody. Thank you, Dean Chang, for this wonderful introduction. And it's my great privilege to stand before you today to deliver this lecture. And I want to begin by expressing my deepest gratitude to Carol and Alan for your vision, generosity, and also legacy, which have made this professorship possible. Alan was a remarkable individual whose impact on the innovation of medical devices and biomedical research really lives on, not just through this professorship, but also through the countless lives that he touched and saved by his devices, and also the innovative work and kindness and passion. So it is a profound honor to carry his name and to continue the work that he so deeply valued. So my lab studies both pulmonary bioengineering and also cardiovascular bioengineering. So today's talk will be primarily on the pulmonary side. So let me just stand here. So the title is Using Cells and Materials to Fight Lung Diseases. So first of all, I think as we're into this stage of the program, maybe you're a little bit tired, so take a breath. Take a breath, right? And every day you take about 20,000 of breath, and actually you would find this is so easy and so relaxing. But there are many people who cannot breathe that as easily as you and I do. And the struggle of breath is a struggle for life. There are actually more than 500 million people who in this world living with chronic respiratory diseases. So if you're looking at the top 10 disease killers in this world, actually 4 of them are respiratory related. Many lung diseases, such as pulmonary fibrosis, and specifically idiopathic pulmonary fibrosis, are chronic and ultimately fatal. So when you can't breathe, nothing else matters. So talking about this particular disease, IPF, which is idiopathic pulmonary fibrosis, on the left side you will see a healthy lung. It's a CT image. So what you can see here, the darker area are actually the good area, which are they filled with oxygen. That's why they show a hypointensity, which is dark in this image. But other tissues, such as solid tissues and like bones, will show a bright color here, like the rib cage and the spine will show a bright color as you can see from here. Cannot use my cursor, but you will see the bright spot here. So this is on the right side. Now is the IPF lung. As you can see, it has a honeycomb structure. There's no bones growing into this lung, but there's fibrosis, which is the dark area, which previously could be filled with oxygen, is now replaced with fibrosis. So IPF is a very significant disease. As you can see, 5-year survival rate is about 33%. And just putting into a context of why we should care about this disease is in the context of cancer. Many cancers today have a better survival than IPF. Looking at how much we can do with breast cancer, 80%, bladder cancer, 80%, prostate cancer, almost 90%. The only 2 cancers worse than IPF are actually lung cancer and pancreatic cancer. When making things worse, now unlike cancer therapies, you only have 2 FDA drugs available to treat this disease, which are nintedanib from BI and pirfenidone from Genentech. They cause GI toxicity. Many patients cannot stay the full regimen, and also they do not work very well. When I say they do not work well, actually you can see this graph. So this is a paper from New England Journal of Medicine, published on the right axis is the forced vital capacity, which is lung function. It's the very single best indicator of pulmonary function, which is forced vital capacity, FVC. And on the x-axis is the weeks. So as you can see, a patient can lose, continually going down and can lose about 250 cc per year when you have this disease. And unfortunately, you don't have much 250 cc to lose because you may start with a baseline of 2,000 cc, right? So this is how the best performer drug does. It neither stabilizes disease nor reverse the disease, but only slow down the decline a little bit. So, and obviously, as someone working in the bioengineering field and regenerative medicine, we like to seek for alternative solutions other than pharmacological therapies and stem cells. So what are stem cells? So stem cells are primitive mother cells that can undergo self-renewal and can differentiate into daughter cells that can make a specific tissue, such as lung tissue, alveoli structure, can secrete pro-regenerative factors, and they are a very important part of regenerative medicine. So in the last decade, we developed this technology called lung spheroid stem cells, which you can derive billions of lung stem cells from either autologous cells, which is from your own body, from a small biopsy, or allogenic sources from donor lungs. On the left side, you will see they are not grapes, by the way. I know you are eating some fruits, but they're not grapes. They are human alveoli structures, right? They look like this is a 3D model of alveoli structures, which is the structure that's responsible for gas exchange. And they are wrapped with vessels. You see the veins and small veins and arteries. On the right side, they look like alveoli structures. They are the cells that were growing in the dish. And in certain conditions, they will differentiate and become those structures that resembling a human alveoli structure. So we call them growing a regenerative organoids or vineyard in the dish. So these cells, they express stem cell markers. So we use kind of surface markers to make sure quality control their stem cells. They are also containing supporting cells, as you can see from these different markers. I'm not going into details, but they're expressing a mixture of stem cell and supporting cells. So now before you can go to human trial, you have to do some sort of preclinical study. And usually we hire volunteers, normally are rodents first, preclinical studies. So on the left side, you will see a perfectly healthy rodent lung, mouse lung. It has a pinkish color, has no problem at all. And on the middle side, it's a PF, which is pulmonary fibrotic mouse lung. You will see the darkening of the color, which is indicating fibrosis. And certainly, mouse, they do not develop IPF, so you have to give a certain a drug to induce pulmonary fibrosis. And this is, on the right side, is when we inject these human lung stem cells to the specific mice strain, you will appreciate that the lung is now looking much better and actually resembles the healthy lung. Now, after many, many years of work, you have to do a lot of safety and efficacy studies to prove submit a like 400-page document to FDA to convince them that you can do a clinical trial. And then we started a trial which is called a HOUT IPF trial, which is a human autologous lung stem cell therapy for IPF, and in a short name, acronym of HOUT IPF. So we take a small biopsy from the patients who had IPF. They come to the clinic. The procedure takes 15 minutes. It's outpatient clinic that's sending a biotome to take a small piece of tissue the size of a rice. And then the cells, the tissue will transport to our GMP, which is good manufacturing practice, cell culture lab, which is similar to Carol just told this morning in my lab. And then these cells will self-aggregate into those lung spheroids, or you can call them miniature lung cells in the dish. And then in a final prep, we prepare a single cell solution in saline that could be intravenously injected. And again, this can be done in a IV infusion center that will go back to the patient's body and go to the lung and migrate to the area of the lung and cross the barrier of the blood vessel and go into the fibrotic area, regrow the healthy lung tissue, but also chewing up the fibrotic tissue. So replacing the fibrotic tissue with healthy new alveoli structure. Now, I'd be pleased to, the paper hasn't been published yet, but I'm pleased to show you some data from the first 9 patients. Looking here is a black line, so there's 3 groups. The first 9 patient data, the black line is a control patient which received standard of care, including those drugs. Low dose, it received 100 million cells, and high dose received 200 million cells. And by the time of 12 months, we lost about 60% of our control patients. They either passed away or had to receive a lung transplant. But actually, lung transplant is also very difficult. There's a very long waiting list. Many patients could not benefit from lung transplant. And this is the patient received the cell therapy. We still have 100% survival. Now, it's a number game. The same cell that we're using for the previous clinical trial, actually is ongoing clinical trial, you can make thousands of exosomes from one stem cell. And what actually is better is that now you can inhale these particles instead of injection. So there's a possibility of self-administration and repeated dosing at home versus you have to go to a infusion center to get your stem cells. So actually here showing a imaging data that after inhalation, actually take you a tour deep into the lung, you will see that these particles, these exosomes are labeled with red color. You will see they are nicely distributed all over the place in the lung, across the barrier of the airway. Other than acute lung injury, and actually a lot of acute lung injury can be caused by infectious diseases, such as COVID. I think 10 years ago, nobody cared about this picture, but now everybody knows this picture, which is spike protein. You know, spike protein on the SARS-CoV-2 virus docks on the ACE2 receptor on our lung cell. So it's like a key, open the lock on our lung cell, and they can enter the lung cell and start to do all kinds of bad things, release the genetic materials and to replicate themselves, and then really cause huge lung injury. So taking a similar approach, we are using the same stem cell. We find that there's a naturally highly expressed AC2 on the stem cells that we're using. So we kind of making those nanobubbles again from these cells. They serve as drones. And once inhaled, they will actually capture those viruses and saturate the spike proteins on the virus. So we call them a nanodecoy strategy to saturate the spike protein. So this is actually primate data. We see that on the blue dots are the control animals, and then the red dot are actually inhaled with nanodecoy. And you will see that the y-axis is by log viral load. In day 4, starting day 4, you don't actually, there's no measurable detectable virus in those animals received this infusion. And actually the lung structure looks also much better. And we actually received another approval from FDA to use this lung stem cell-derived secretome to treat patients with acute respiratory distress syndrome, which are actually the disease with very high mortality and actually killed most of the COVID-19 patients. Well, luckily, we don't have many of those severe COVID-19 patients in the ICU right now, but this therapy will always be ready if next time we have another pandemic by COVID-19 or by something else. Now we talk about all these lung diseases, IPF. Some of the numbers are accumulative number of patients. Some of the numbers are annual patients, like COPD, which is chronic obstructive pulmonary disease, 11 million. ARDS, which is the acute lung disease. Viral infections, we have accumulated. This is 2022 data, actually. More than 100 million people get COVID-19. And also pulmonary hypertension. Those are just numbers. Silicosis, which are from pollution, from people working in the mine, they have black lung disease. But we haven't touched the one disease, which is also very, very important, which is lung cancer. Every year, more than 230,000 patients in US were diagnosed with lung cancer, and it's a very deadly cancer. So we're using these nanoparticles from those cells to tackle on lung cancer as well. So here, what we did is we load the nanobubbles with a specific drug, which is IL-12 mRNA, which is mRNA is many of us will receive the vaccine. So this drug, when it's released, it actually can recruit the immune cell, the body's immune cell, to start attacking the lung cancer cells. As you can see from here, the black line, these are animal data. The black line is the survival, and by day 30, you lost all the animals with the placebo treatment. The ones that received the exosome with IL-12, the blue line, you still have a considerable 50% of survival. So we're translating this therapy as well. As you will see, this the next graph is showing the lung tumor growth. The blue line is not very much noticeable because they are very small tumors that is really showing the efficacy of this therapy. So now I'd like to actually acknowledge Savannah, who is also recently was with us today, and she had the privilege to be named as a new Kaganov Fellow. So thank you again very much, Carol, for your generosity. I'll talk a little bit about Savannah's work. So Savannah's work is working on how do we make the drugs better. So we're talking about the drugs not working very well. The reason they have such a high GI toxicity is because you swallow them. You swallow them is oral delivery, and it hits everywhere. So that's why it upsets the GI system and causes liver toxicity, is because it's we so-called systemic delivery. So if we can convert this oral drug or intravenous drug to an inhalable drug, which is targeted to the lung, then the toxicity should be largely reduced. But you have a big obstacle. Looking at the graph here, it has something called mucociliary clearance. It's the Mother Nature designed our system to clean our airway all the time. So we're looking at this movie here, showing the particle constantly moving and being expelled by our mucociliary system. And it's actually going upwards. That's why you cough. You're coughing out, and that's why sometimes you also swallow the things. It's a defense system. But this defense system make those inhalable drugs do not work, because you constantly, rapidly clean up and wash out this drug, the drug that you're given. So, and one day, Savannah was thinking about this idea that, just like the hair, if the hair without bundle under the wind, you will go everywhere. It's very fluffy, cannot retain anything. But if you have braids, it stays when there's strong wind. So actually, we actually named this technology braid, which stands for bioadhesive retention adjuvant for inhaled drug. And actually, the idea is that you can inhale a hydrogel, a sphere, that can form a thin layer to reinforce and make your mucus transiently and slightly more viscous and more sticky for the drugs that you inhale. And these are the data. On the left side, you will see the control, the mucus continuously moving. And on the right side, you appreciate that with this inhalable adjuvant, now the particles are not moving one direction, but they can only do Brownian movement, which is good, because once the drug cannot be going upwards, they can only go vertically, which is go to the lung. So it's comparable to the oral dose. Why do we do this? Now, I mentioned to you it's toxicity. Looking at this experimental design, inhalation, 3 dose, 30 milligram per kilogram. The oral dose, 30 dose, and 300 milligram per kilogram. So this is, ladies and gentlemen, this is a dose reduction to 1% that is being used on patient. If we can reduce the drug dose to 1%, think how many patients can benefit without toxicity, GI toxicity and liver toxicity. So obviously, I have to thank everybody in my group who actually, did all this beautiful work, and our funding agencies, and like also take this a moment to thank individuals here. Actually, those are my trainees. I just highlight a few more trainees, on top of Savannah. They went to different institutions, winning different awards, being independent PIs, and some still stay here with me. Some followed me here, and also I started mentoring new physician scientists here, including Christina, who is a very brilliant pulmonologist here at CU IMC. And as I come to this end of the lecture, today, before I came to this dinner, I just finished a 85 student class in the Chandler Hall. Actually, the final presentation. When I was crossing the campus, from the Chandler Hall to this side of the campus, I was thinking, why? People also ask me, why do you move here to Columbia? And I think environment is very important. As I crossing the lower plaza, environment is very important, and we have a top-notch environment here. But ranking is not everything. The key thing is really the people here. And there, I need to really give credit to the many people here at Columbia who helped me get here and support my research. Many of you that I could not highlight each of you on slide, but I'd like to highlight a few individuals. I would like to once again express my sincere gratitude to Dean Chang for your remarkable leadership and support. Engineering for Humanity has also been a mission of my research. And I have to mention Mary. I know that despite you were no longer the dean and provost when I moved here, but you set up this foundation that actually attracted me here, this nice environment. So thank you very much, Mary and Dean Chang. I also like to thank Kem Liang and Gordana Vonyak Novakovic, who are really the two giants in my department. My accomplishments are nowhere closer to what they have done to our field. And I was reading their papers when I was a graduate student. So what a privilege. Now I can work, I'm working side by side with them in the same department. And I really have to thank them to lend their shoulders for me to stand. And I think I will make sure to pay forward to allow young generation scientists to stand on my shoulder as well. I'd like to thank my current and past department chairs, Paul Scheider, who is with us today, Ed Gore, who is not here, who are terrific department leaders. As you can imagine, it's not easy to move a big team out of state. And they really have been so supportive and understanding, has really a can-do spirit. In these days, it's really much easier to choose can't do over can do, because can do means risk taking, and can't do is much safer. But research only thrives with can do spirit. So I'd like to thank my department chairs. And also many people that I could not put it on the slide, including today in the audience, and Helen, Irene, Buckley, who are our engineering leadership, who are taking care of me, talking about faculty housing, to kids' education. Every single factor they're helping me and fix it for me so I can be focused on my research. And also this wonderful team today who putting together this show and helped me. I cannot name all of them, but Keely, Doreen, Kalinda, Cody, thank you very much. But lastly, but mostly, I like, again, thank Carol and Alan. Words cannot fully convey my appreciation for your incredible generosity and the gifts that made this moment possible. And this professorship and everything it represents is a testament for Alan's legacy. And while Alan may no longer be with us today, I strongly believe one day I will see him, and I wish he will be happy for what we will have done for treating those incurable diseases. His vision will live on not only this professorship, but will also be reflected on every single paper we publish, every single student we will mentor, and every single innovative clinical trial that we will launch. And it is my privilege to carry his vision forward, and I'm so grateful for both of you for entrusting me for this responsibility. And thank you very much.
So can you hear me? Great. I think we have time maybe for 2 or 3 questions. And for those of you who are still thinking about questions, please send this a QR code on the program. You can scan that and send your question. We'll make sure we'll give it to Professor Chen here. So we have a few questions here. Let me just choose one. Maybe the shortest one. Is this the technique you mentioned so amazing and really so compelling? Is any of them applicable to cystic fibrosis?
Yes, and actually, Ding-Chen, have a seat, please. Yeah, I will also stand if you keep standing. So thank you, Ding-Chen. So I think there's a lot of potential. And this CF is definitely also on our radar. And CF is one of the few diseases that has a very clear gene target, the CFTR gene that is faulty, that makes actually sicker mucus. So actually, we had some. Okay,
no,
all right.
That's okay.
Yeah.
I can ask from here.
All right, so, okay. And actually,
okay. Now you can use
Yeah, okay, all right. So, thank you. So CF is definitely a disease on our radar, and we have shown that by inhalation of exosomes, you can deliver a specific mRNA to address the specific gene mutation that CF patients have, which is CFTR. And also, potentially, we can also deliver CRISPR therapies with these little stem cell nanobubbles. So all it does is drug delivery. Drug delivery is very important in the space of lung diseases because it's very hard to cross the barrier, which is the mucus. Yeah.
Fantastic. The next one from Ming Ma. Using inhalation, how do you optimize targeted delivery of this therapy to effective lung areas?
Yeah, this is a great question. So again, drug delivery issue. So first of all, doing a nebulization or inhalation, you're already winning at the start line. It's so much better already, better than intravenous, which intravenous hitting every single part of the body. But in inhalation, really, you already target on the lung. Now, the next hurdle would be how do you cross the mucus barrier, the epithelial barrier, and also hit the cells that you want to target. And there's certain ways that we are doing in the lab, which is engineering the surface of the stem cells and the stem cell nanobubbles that are targeting, that actively can find the region or the foci that needs to be fixed. So it's something called a surface engineering or a stem cell engineering that you can, just thinking about it, you're adding GPS on the surface of the cells and nanobubbles so they can find the way to the place they have to go. Yeah.
Great. Last question. We have more than 10 questions here. We'll make sure I give it to Professor Chen and then provide answer to you. The last one is interesting. What inspired you to conduct research in this area, biomedical research, from beginning in the first place?
Yes. So when I was a child, the passing of the first family member is always very striking to you, right? So my grandpa passed away when I was an elementary student from, he works in a mine, and that at that time, there's a lot of dust from the mine, and there's not much occupational protective equipment at that time. Probably he didn't even wear a mask. So it's actually silicosis, which is lung fibrosis. It's very different from idiopathic pulmonary fibrosis, but this is another type of pulmonary fibrosis, which is a permanent deposit of those dusts in his lung, and actually killed him. And this I was a child, but it was, when you was first time in a family member's funeral, it's give you very, very huge impact that someday I like to work on this disease. And I started my training on cell therapies and on cardiovascular diseases, and then later on started working on pulmonary fibrosis, which is really with a very slow of federal funding, but really has to be addressed with an unmet medical need. Yeah.
Thank you. And what you're working today is so meaningful. We bring so much impact. Thank you. Let's
Thank
thank Professor Ke Cheng again.